last but not least, our last speaker is Rishi.
Hi guys, how you going? I'm Rishi. I'm a PhD student at Uni of Sydney. I'll be talking about work I've been doing as part of my project with my supervisors Eli and Ben. I'll start with some background to my work. So we're interested in understanding the structural mechanisms that give rise to the patterns in large-scale or macroscale brain dynamics that we see in real measurements. And I'll be focusing specifically on that of the cortex. And in the cortex, structural connectivity is largely distance dependent, meaning the chance of a connection between two populations tends to decay as a function of the distance between them. And it can be shown with some assumptions that activity propagations through this form of connectivity is largely determined by the local geometry of the cortex. So I'll call it local connectivity. There are also exceptions to this rule where there are these specific long-range connections that connect remote populations on the cortex. And although spatially aggregated, they do still follow this distance-dependent rule, but only conditioned on the positions. And they are non-uniformly positioned in space, so they're not completely accountable by this local geometry. Now, both of these structural mechanisms are simultaneously shaping cortical dynamics. I'm just showing here an example of how a mouse cortex responds to a whisker stimulus measured with voltage-sensitive dye imaging. So you can see how the red dot is initially activated. There's a traveling wave from it due to the local connectivity, and then the blue dot is activated before the wave reaches it, and that's coming from that local, the long-range connection from the red to blue dot. Now, despite clear evidence like this, where you have both structural mechanisms shaping the dynamics, recent results have indicated that many patterns of macroscale brain dynamics can be captured by just considering a local connectivity rule that smears out the specificity. Examples including resting state fMRI most compellingly, but also in resting state MEG and task fMRI activation maps. Now, from a modeling perspective, this is good news because part of the game is we're trying to build models that are very simple ingredients but can capture a wide array of phenomena. And here we just need the shape of the outer cortical surface and do not need any connectomic data of the actual connections between specific populations. But it's not clear yet as to why this is the case. Why could you just take a local connectivity and capture a wide range of phenomena? Reasons being that we know that these non-local long-range connections facilitate fast information processing, such as the whisker stimulation example. And we also know that they're very clinically relevant. We see that they have specialized functions from neurological experiments from the last century. They also are pathways for seizure propagations in epilepsy. Put together, the question we want to pose is, why do these long-range connections appear to have a relevance in some dynamical settings, but not in others? And we tried to pursue this question using a simple computational model that we constructed. So essentially how this model works is you have this cortical surface, it's shown by a square here. It has a local connectivity, and then there's specific points on this surface that are connected by long-range connections. And the way this model works is if any stimulus arrives at the surface, the local connectivity will facilitate a traveling wave from the stimulus site, and then these long-range connections act as these wormholes that teleport activity between specific locations. And here's an example I'm showing with just 50 long-range connections. Moving on to our results, we first investigated how these long-range connections influence the dynamics across different timescales, ranging from the millisecond timescales inherent to fast information processing to the slower orders of seconds timescales resolved by resting state fMRI. And we find that the influence that these long-range connections have on the dynamics are largely timescale dependent, and they have the largest influence over these short millisecond timescales. Just show a simulation here. If you compare the evoked responses with just local connectivity and with the extra long-range connections, you can see that the influence of the long-range connections is strongest at around the 20 millisecond mark. And the importance of this result is that it suggests that these long-range connections can influence the dynamics most prominently over these short millisecond timescales. And in contrast, if you restrict your dynamics to these slower timescales, for example, in fMRI, you have the hemodynamic response of the two models shown by the thick horizontal line. We can see that the long-range connections are still influencing the dynamics, but this influence is much less prominent than when you observe over the short timescales. The second thing we looked at is how do these long-range connections influence dynamics depending on where the input is positioned and how precisely it's positioned. Motivation behind this is we know that these connections are non-uniformly positioned in space, forming hub regions, and these hub regions can also be tightly interconnected to form rich clubs. What we find is that when the long-range connections are homogeneously distributed, so in a random sense, in the left column, the influence is also largely homogeneous in space. However, when we add these non-random effects through either introduction of hubs or rich clubs, the influences tend to be more non-uniform in space, shown with the middle and the right column. And if you compare this with the random connectome over an ensemble, we see that two things. If it was possible to apply a precise stimulus, let's say a hub region, you will induce very large effects on the large influences on the dynamics. And this would have to be likely done with some external targeted stimulation procedure. However, these very spatially localized influences at the hub regions are somewhat more washed out in spontaneous settings where there is no explicit stimulus applied to the system. Reason being that the hubs form a very small proportion of the entire cortical surface. And so we demonstrate here that the level of influence of the long-range connections are also interestingly tied to the precision of input that is actually applied to the cortex, either through externally or through thalamic control. Particularly if you have a large influence, particularly that you have a large influence where you're able to apply stimulation precisely at a particular location, but less relevant when you're observing it in a spontaneous state. And so coming back to the question we aim to pursue, our results now paint the spectrum that can possibly explain why the influence of long-range connections on the dynamics tends to vary with the setting that the dynamics is generated and measured, starting with stronger influences on the fast processing of precise sensory stimuli, but weaker influences on these slower, spontaneous fluctuations. And these findings are somewhat consistent with experimental findings and thus may reconcile the disparity in the findings on the relevance of long-range connections. I'll now move on to the implications of these findings. These have implications on conflicting perspectives on how best to build mechanistic models of macroscale cortical activity. In the prevalent connectome view is that cortical activity evolves on a network of lump nodes representing parcellated regions, and the agents between these nodes are an accumulation of fibers, both local and non-local. However, the contrasting perspective that arises from neural field theory is that the evolution of activity governed by just a local connectivity rule that smears out the specificity on a continuous cortical surface is a sufficient approximation of real cortical activity. And our results suggest that this neural field approximation becomes a stronger approximation for capturing the spatiotemporal patterns of spontaneous dynamics on longer timescales, like that of resting state fMRI, whereas the additional non-local specificity of the connectome becomes more crucial for capturing the cortex's more rapid processing of spatially localized stimuli. This does not imply that the specificity of the connectome is not important for capturing resting state fMRI dynamics, rather that our work results suggest that emerging targeted stimulation technologies, which can measure stimulus-evoked responses at fine temporal resolutions, as an experimentally challenging but an important regime for developing and refining future dynamical models of macroscale activity informed by connectomic data. Reason being is we believe this regime is in which the specificity of structural connectivity may play a more significant role than in resting state fMRI. Examples include optogenetics or transcranial magnetic stimulation, combined with modalities which can measure the millisecond timescales at which these responses evolve over. And to that, I will conclude. Thank you to my supervisors, colleagues. Here's a link to my poster and paper. I'm happy to have a chat over this. There's much more work. It's a very simple model, of course. My poster's at 1440. Thank you all for listening.
Thank you so much. It's nice to see like the field and the connectome coming together. Is there any questions from the audience? Yeah, go ahead.
Yeah. Hello. Yeah. Okay. So, thank you so much. Really interesting talk. The question you addressed in this study was the one that I actually really wondering or wanted to investigate myself. A beautiful finding. My question is, so one of your main finding was the influence of long-range connection made a peak around 20 to 30 milliseconds. So that's how you conclude the influence of long-range connection is mostly early time, like a shorter time scale, right? As a neuroimager who was working on whole brain, large scale, for instance, default mode networks, they're relatively far, like 10 centimeter far across the whole brain. Have you ever expand the scale of your modeling, and then if the distance is far more than what you model along this local manifold, still your findings holds?
So I think the distance between the remote regions does have an effect on the correlation structure. There is reason behind that. It's largely to do with the specificity of the input driving the system. We can chat about that afterwards.
It's just to me, like my example is default mode network. They are relatively far each other, and I just wonder whether your finding is exactly applicable to this much far distance of the distributed networks, for instance. But we can talk later.
Yeah, so I don't exactly can't hear the question, okay. So we can talk later. Yeah.
Sure.
Yes.
Yeah, thanks for the question, and thanks to Rishi and all of the other speakers.